I have a test tube here. The label of this test tube says chemical X. Can you see anything inside? Can you see chemical X inside? Take a look at it. Can you see anything inside? Let me invite one of the students here. Why don't you come up here? Can you see anything here? Can you see anything here? See, actually, nothing is in here. The easiest way to see the chemical inside is to look for its shadow or color. That's why you do it like this. If you can see the shadow or color, there is something in here. Because many chemicals absorb light. That is the definition of spectroscopy. The light is actually a wave, electromagnetic wave. The radios and microwaves have long wavelengths, while X-ray has short wavelengths. The shorter the wavelength is, the higher the energy is. In other words, the energy levels are proportional to the frequency. Our eyes can detect only this part of the light. We call visible light. And ultraviolet light is absorbed by many chemicals and useful for chemical characterization. For example, DNA bases absorb ultraviolet light or UV light around 260 nanometers, while peptide bonds absorb UV around 220 nanometers. Aromatic residues, tyrosine, and tryptophan absorb UV around 280 nanometers. By the way, the single letter codes for tyrosine and tryptophan are Y and W, if you remember. If your protein has tyrosine or tryptophan, your protein should be able to display this kind of UV spectra, right? On the other hand, DNA should be able to give you this kind of UV spectrum at 260. Different chemicals absorb lights at different wavelengths. For example, I have 3 pieces of papers here. The white paper does not absorb light. That's why it is white. Black paper absorbs light at many different wavelengths. That's why it is black. Yellow paper absorbs the light except yellow light. That's why it is yellow, right? So then, okay, different chemicals absorb lights at different wavelengths. Then what's going to happen after chemicals absorb the light? Light absorption increases the energy level from here to here of the molecule to its excited state. The energy is usually released gradually as heat through the vibration of the molecule. That's why white materials do not get hot, but black ones get hot by absorbing light and emitting heat. Some chemicals release the energy by emitting light instead of heat. That is fluorescence. When a fluorescent chemical is excited by absorbing light, energy is released a little bit, and then the molecule emits the light back. Since the energy level is slightly lower here, the emitted light has a smaller frequency, which means a longer wavelength. This difference is called Stokes shift. As a result, the fluorescent molecule is excited by a light and emits a light with a longer wavelength. Here is a typical fluorescence spectrum. This molecule absorbs light right here. This fluorescent molecule absorbs the light right here, but emits the light at a slightly longer wavelength. Then, if the wavelength of the emitted light is within the visible region, you can actually detect the emitted light by your own eyes. These are the structures of well-known fluorescent compounds which emit visible light. DAPI emits blue light. The fluorescein emits green light. Texas red emits orange light, and Psi5 emits a red light. A number of fluorescent molecules are quite useful in cell biology. I will show you two examples. Here is a structure of WHITS 33258, which binds to DNA, and thereby labeling the nuclei of the cells. You can see the nuclei of the cells very clearly in this picture when labeled with this fluorescent molecule. The leftmost cell right here is in the metaphase stage of mitosis. So its chromosomes emit light brightly because they contain highly compacted DNA. So you can follow the cell division by looking at a DNA labeled by fluorescent molecule. Another example is a mitotracker red. The structure is right here. This is a popular fluorescent probe that labels mitochondria inside the cells. These red tube-like structures are mitochondria, and the nuclei as shown in blue. The different fluorescent molecules emit different lights. Then they are sometimes useful in the cell biology. In the next video clip, I will show you another example of fluorescent probes from our own research.